Hey guys, I'm Dipanjan Ghosh. Today I want to discuss the need-based discussions of biotechnology and very few questions from biotechnology which are very relevant in now for NEET exam, such as who have discovered biotechnology, types of restriction endonuclease, the cutting site of EcoR1 or Hind3 or Hind2, or the discover of first restriction endonuclease, or the different types of vector such as expression vector or transcription vector. So now the time of discuss of that particular portions. So here discuss about that topic. So the first questions, which is very relevant in now NEET examinations, that portions that is biotechnology. So biotechnology, that terminology is actually first proposed by Karoye Eriki or Kai Eriki, Erkey Eriki in 1919. So all of you have to keep remember very important the terminology first proposed by Kai Erkey. And types of restriction endonuclease, all of you know. Restriction endonuclease, we do its function to a site which is known as the restriction site or the recognition sequence. Restriction site. And there are very few types of restriction, type 1 restriction endonuclease, type 2 restriction endonuclease. Basically, they have cut the sequence to a very specific distinguished site. And types are basically four. Four types of restriction endonuclease have been found. And there is a questions to the NEET examinations. So what is the first restriction endonuclease discovered? So EcoR1, Hind2, Hind3, AVA1. So EcoR1 basically we know the sequence 5-GAATTC. So 5-GAATTC, this is the recognition sequence, and the restriction site is the 5-G end we have seen. So we have seen here, and the source is the Escherichia coli RY13, and the cutting site are sticky or cohesive. Hind3, in case of Hind3, the source is the Haemophilus influenza RD, and the sequence is 5-AAGCCTT, 3 It cuts to the sticky end. Hind3 to the sticky end. And AVA1 we have seen the cutting site is the 5'C. So this is the purine and this is the pyrimidine. This denotes purine and this denotes pyrimidine. Okay, so that portion is the cutting site. And source is Anavina variabilis. Okay, and it cuts to the both sticky and cohesive end. And now I want to discuss the answers. Which will be the answers? The first discovered that is the Hind2. It cut to the 6 base pair sequence, and the recognition site is the 5-GT pyrimidine purine AC3 So this is the cutting site I have seen here. Okay. So the answers will be Hind2. The answers will be Hind2. Okay, and now I come to the aspect that is the vector. We know vector is nothing. It's a vehicle, where we can incorporate the gene. So we have seen here, I have discussed now the expression vector and transcription vector. Expression vector means where the transgene is actually expressed. Here the transgene is actually expressed. Now the question is, what is transgene? What is transgene? What is transgene? Now whenever we want to incorporate one external gene to the vector, then it will call the transgene. And it is expressed to the host cell, so that's why it is called the expression vector. Okay, such as for example PGX 3x plasmid. And whenever it will not expressed, only the transcription is possible, then it is recommended as the transcription vector. So for example PBR 322. So thank you.